Alright, in this video we're going to look at the law of independent assortment. Alright, so what Mendel was able to do, alright, sorry, this should be an E, okay. What Mendel was able to do with the law of independent assortment was he was able to show that traits on different chromosomes, all right, are not required to sort together, but they can sort independently. All right. So the basic idea is that, again, if we show up, right, if we think of each parent, right, or each cell, right, so we've got two chromosomes here, all right, or two chromatids of a chromosome, all right, and we've got two maternal, right? On these chromosomes, we can put some characteristics. So we can just make this one here to be, right, so we'll make this our location, all right? And let's just say that this is a heterozygous individual for seed color, yellow versus green, and so we have capital Y on the paternal, okay? And we have lowercase y, or green, on the maternal chromosome. So the law of segregation would say that we have to separate these different alleles, right? That we have to separate these out. Now, in terms of independent assortment, though, we're looking at two different things. And so let's go back and draw another chromosome. All right, and I'm going to draw these down here just to illustrate our point. All right, so we've got our maternal up here this time, our paternal on the bottom. Okay, so this is all still one cell, right? And we're in meiosis 1. All right, and in this case, we want to add, we're going to add seed shape to this. All right, and so this is our round. Oops, hang on. Do a little chromosome repair here. There we go. All right. Now, what we want to notice is that, so if we had this individual, right, and if it was completely heterozygous for this thing, right, it doesn't really matter where those where the alleles have come from, all right. But what we want to illustrate is that we could have the round down here and the wrinkled here. Okay, so let's say that when we generated this individual, we had a homozygous recessive with a homozygous dominant, right? So the male would have been homozygous dominant, the female would have been homozygous recessive, just to generate something that looks like this. Now, what this is showing us, though, is that if over here on the left we have color of the seed, all right? And over here on the right we have shape, all right? Now, these things can flip-flop, right, in how they line up, right? Either of them could do this. So we can get different combinations of this. So what that is saying is that you don't have to segregate the color that would be the dominant color with the dominant shape. All right? You don't have to move the wrinkled shape with the yellow or the green color. That's not a requirement. You can do it either way. And so what it's saying is that these chromosomes here align, okay, independently of how these align. And this allows for new combinations of traits. All right, so we're going to find new combinations of traits, new combinations of alleles. All right. So what Mendel did was he started out with a P generation. All right, and he took true breeding. All right, so big Y, big Y, big R, and he crossed that with a true breeding recessive. All right, and so this would be on the left, our homozygous dominant would be yellow and round seeds, and on the right we would have wrinkled and green. All right, so from here, all right, each of these can only generate one type of gamete. All right, so we get one big Y, big R. Those are all the only options you can get. And over here you can only get little y, little r, right? Those are the only options you can get. All right, so we combine those together and that will give us our F1, which is big Y, big R, little r. This is our dihybrid. Okay, now our F1s are dihybrid for two traits. Okay, now at this point I'm only going to move this forward for independent assortment, right? So we got independent assortment. Okay, so if we look at taking our F1 and crossing it, right, and we're going to cross that with another F1, okay, we're going to try and generate our F2, all right? So let's do the Punnett square, okay? Now, let me let me shrink my Punnett square down here a little so we can have a little bit more room, okay? What we want to do is we want to make every gamete possible. So this is my female, this is my male, okay? So let's start with the first allele of our color. All right, so we've got yellow, so we're going to combine that with this, big Y, big R. Now let's do it again. So we've done inside, outside, right? So we've done big Y, little r. Now let's do the insides. So we have to start with what are the combinations for this second color allele. All right, so we've got little y, big R. And then we'll take that one all the way out, right? So we've got these two combinations, and now we have little y, little r. Okay, let's do the same for the male, make some sperm. So we're going to go big Y, big R. All right, we're then going to go big Y, little r. That's our other combination for big Y. Now let's look at, oops, this should be little y. Now we take our little y allele and we combine that with big R. Okay, and then we can go little y, little r. And we have all of our possible combinations. Now, if we take all of this stuff and we combine it, all right, we will end up, right, so we're going to combine them like so, right? Okay, and you can fill this out the rest of the way. Okay, you can think of this and do this, I would hope. Now, what we want to look at is what happened, right, in terms of the results. And so what Mendel ended up with were, he ended up with 4 phenotypes. He ended up with yellow, which is our dominant, okay, color, and round, which is our dominant shape. And let me capitalize this just so that we can, all right, so we get yellow round, all right, we can get yellow wrinkled, okay, we can get green and round, and we can get green and wrinkled. Now, what Mendel observed was he got 9 of the dominant, 3 of these new combinations, and 1 of the homozygous recessive. So this is a parental phenotype. This is also a parental phenotype. Okay? These two, however, did not exist in either parent. Okay? And so that generation of new phenotypic combinations suggests that how the yellow-green alleles, all right, assort, and how the wrinkled round alleles assort, all right, is not dependent on each other, meaning that you can get a yellow plant, a yellow seed that is wrinkled. You can get a green seed that is round. All right, even though the parents, the original parents, all right, the P were yellow round, green wrinkled. All of the F1s were yellow round, all right, and now we have these new types. And so what that is suggesting then is that you've got 2 chromosomes. And I'll make one big, one short, okay? And on this chromosome we have color, and this is a diploid individual. And on this chromosome over here we have shape. All right? And this is my heterozygous individual. The gametes from this, right, can end up being big Y, big R, big Y, little r, little y, big R, little y, little r, right? And these are our gametes that we can get. And therefore we can get new combinations, all right? All right, so that is the law of independent assortment, which states that each pair of alleles segregates independently of each other pair of alleles during gamete formation, which is exactly what we saw right here, okay? It applies only to genes on different non-homologous chromosomes, which is what we see here. Let's say that this is chromosome number 1. We have 2 copies, right? And this is chromosome number 2, all right? So that is the basis for independent assortment, okay? And what you need to do is, if you're still confused on this, is you need to go through meiosis with those gametes, right? That's the if you're still struggling with it, then you're going to have to do meiosis with those gametes and look at it in terms of what are the combinations you can make, okay?